coming to this event tonight in which we celebrate student research scholarships and expeditions. I have to say, and I probably say it every year, but it's absolutely genuine, that this is one of my favourite events in the university calendar. I know that that is shared with a number of people in the room, but it's so great to see some of the fantastic research and expedition work that our students are undertaking throughout the course of the year. And I think it's really important that we do that as part of our university public lecture series. So many thanks to those of you who are members of the public, no particular affiliation to the university, for coming along to this event and supporting our students and our staff on this, what I hope will prove to be a really enjoyable evening. So I'd just like to say a few things about the research scholarships and expeditions, particularly focusing on the scholarship element. And then I'll hand over to my colleague Emma Pearson, who is the chair of the University Expeditions Committee, to tell you a little more about the history of expeditions in the university, which is fascinating for those of you who aren't aware of it. And it sets a really nice context for some of the presentations that you're going to hear later in this session. You're going to hear, most importantly, from some of our students who've undertaken research scholarships and expeditions during this year. There's a slight change to the programme as billed in your booklet, in that the presentation, which is listed as presentation 5, unfortunately isn't going to take place. But we will have 2 research scholarships and 3 expeditions being presented this evening. At the conclusion of that, there will be an announcement of poster prize awards. Many of you will have been in the Great North Museum this afternoon, looking at the posters that students have produced to demonstrate the research and the expedition work that they've been undertaking. And year after year, I'm absolutely bowled over by the quality of the work that our students are undertaking and presenting and articulating to our judges. After the event here in this building, everybody is very, very welcome to join us at the Great North Museum, so back in the room that we were in for the posters, to have some supper with us, to talk informally with students and staff about the work that they've been doing. So I do hope that people will come and join us for that. You will all be very, very welcome. So we've been undertaking research scholarships in one format or another for more than 25 years. And certainly over recent years, these have been very much opportunities for our students to experience what research is like. And to do that, usually in the penultimate summer of their degree programme. As you'll have seen from the booklet, and if you were in the Great North Museum, a very, very wide range of activity in different areas in the context of this research scholarship scheme. So all 3 of our faculties are engaged in this scheme, and also students and staff from our global campuses, particularly in Malaysia and Singapore. This year we have seen a 30% increase in the number of students undertaking research scholarships, which is a fantastic achievement. My ambition is to increase this opportunity year on year, so this year I'm a very, very happy woman in relation to this. We have a number of funding routes to enable us to do this. there's some university funds which support the research scholarships, and you can see that a large percentage of the research scholarships are funded internally. We've also had very significant success this year in terms of externally funded scholarships. So my thanks to staff and students who've applied for that external funding, and I think particularly for the quality of the research proposals that have been submitted to those external bodies, which has really increased our success rate, which is a really fantastic achievement. So the whole idea of the research scholarships is about our undergraduate students getting an opportunity to do some additional work in that area of research. And all of our students will have selected the type of research that they want to carry out. So looking at the portfolio of opportunities across staff in the university and working with staff members to develop a research proposal, all of which is great experience for all of our students in terms of developing a series of skills and attributes which will stand them in good stead when they undertake some research as part of their degree programme in a formal way, but also when they exit the university and either go into research or into some other employment environment. So this is information about those external research scholarships and the variety of different organisations that have supported about 15% of our research scholarships this year. And so our grateful thanks to these organisations and institutions who have supported our students on this research, and long may that success rate continue. So I shall now hand over to my colleague Emma, who will tell you about expeditions in the university, and Emma will also introduce our student speakers this evening. Thank you.
Thanks. Okay, so I'll just say a few words about expeditions here at Newcastle University. We have an exciting history of expeditions. It dates back to 1948, when a group of undergraduate students went on an expedition to Iceland. And this was under the guidance of Hal Lister, who's pictured here on the left. Hal was a lecturer and later a reader in polar studies here at Newcastle University. He was a glaciologist and a renowned Arctic and Antarctic explorer. And he took part in expeditions, several expeditions, both to the north and to the southern polar regions. A couple of his notable expeditions include the British North Greenland expedition in the early 1950s, after which the team were awarded the Polar Medal, and the Commonwealth Trans-Antarctic expedition, after which some rock heights were named the Lister Heights after Hal Lister. And when he wasn't jetting north and south on all these exciting expeditions, he was a real advocate of his students gaining experience and fieldwork experience through expeditions. He was a real field scientist and wanted students to gain that experience as well. And so that's why he supported students to go on expeditions, first to Iceland and also to Arctic Norway. And this shows a photograph from the first expedition to Iceland with some students using a hand ice corer, which from the inset picture can show that it needs a bit of brute force. Sometimes it's not an easy task doing fieldwork in these environments. And later, after these early expeditions, then he really encouraged students to carry out their own research expeditions. And that legacy has continued today and carries on today. So in the university library there is an archive of all the expedition reports from student expeditions going back to 1948. There's also some really interesting documentation from the North Greenland expedition. And if we look at the archive from the Courier, which you'll be familiar with, the Newcastle Courier, this was from November 1957, when Hal Lister, pictured here, was en route for the South Pole. There was this article in the student newspaper following the first direct contact from Hal Lister with the head of geography at the time, Professor Henry Dayish, during this expedition. And his message was, "Work speeds up a little as anticipates return of sun, but short-handed and apparatus trouble reduce observation time. Hope you received 4 penguin eggs and whale tooth by a London office. Mean annual temperature minus 30 degrees C. How you must envy us in your heat wave." So I'm not quite sure what heat wave there was in Newcastle in November 1957. And also it's interesting to note that this was 2 years before the signing of the Antarctic Treaty. So I don't think our head of geography would be receiving penguin eggs and whale teeth today. So in terms of Newcastle University's connection with earlier expeditions, this is Gertrude Bell. She was born in 1868 in County Durham and has very nicely been described as not your stereotypical Victorian lady. She was an archaeologist, an explorer, a mountaineer, and she played an important role in many expeditions, in particular to the Middle East, archaeological expeditions and mapping expeditions, and also later had a very important archaeological and political role, particularly in Iraq. And the University Library houses the Gertrude Bell Archive. It's a really interesting archive, full of photographs, really fascinating photographs and letters and documents from these expeditions. And in 2017, this archive was included in the UNESCO Memory of the World Register, which really highlights how important it is and what global significance the archive has. So that's a bit about history of expeditions and links with other expeditions. If we come to today, Newcastle University is still very active in terms of student research expeditions. Usually there are about 3 or 4 expeditions that are awarded funds towards their expeditions, and these are from a range of funds within the university. And also there are awards, the Harry Collinson Travel Scholarship and the Sonia Stonehouse Expedition Fund, which were set up by members of their families in their memory, and they were members of staff at Newcastle University. Students also apply for funding from external sources, and this year we had 3 expedition teams who went to Greenland, Peru and Russia, and all 3 of them obtained funding from the Royal Geographical Society, which is an amazing achievement, and also funding from other external sources. And they managed to raise more than £25,000 towards their expeditions, which is an incredible accomplishment. I'm a geographer, so I always like to show a map. I always like to see where our students have been, and this just depicts all the places. The larger the point, the more expeditions that have taken place there. Over the last 72 years, we've had 150 expeditions to 92 different countries. And if we just look at this year's expeditions, so we had one to Greenland, which was the 7th expedition in the history of Newcastle to Greenland. This year was the 2nd to Peru. The last one was in 1984, so I think we're about due to return. And then this year was the first student expedition to Russia. So that's all very exciting. And lets me put an extra point on the map, which is nice. So heading on to our presentations this evening. So we have 5 presentations, 2 from research scholarships and 3 from our expedition teams. So without further ado, we will move on to those. Okay, so first up we have a presentation by Agatha Chu, who is a research scholarship student, and she's going to talk about how does hearing loss lead to dementia.
So hello, good evening everyone. I'm Agatha, a third-year medical student. The title of my project is, How Does Hearing Loss Lead to Dementia? So firstly, why do we even bother researching about this? Well, that is because hearing loss in midlife is the strongest risk factor for dementia. And currently, we do not know the reasons behind. There has been a few proposed theories. So for example, hearing loss may lead to people withdrawing socially. That may increase the risk of getting depression, which can accelerate cognitive decline. But it can also be that there are brain structures that are responsible for hearing, which may be directly linked to cognition. Now, the key message from this diagram on the right is just that if you have hearing loss, you're at 5 times more likely to have dementia, and therefore this is a big deal. So the second question is, what is hearing? Well, there is hearing in quiet and hearing in noise. So hearing in quiet is traditionally measured by the pure tone audiogram, whereas hearing in noise relates more closely to real life. So for example, if we all imagine ourselves now at a very noisy, busy environment, or let's say we're at a party and we are trying to engage in a conversation with a friend, we will need to be able to pick out the friend's voice. So we have to pay attention and also remember the sound, and at the same time inhibit any background noise. So this process requires a lot of cognitive mechanisms, and we think that hearing in noise may provide a strong link to cognition. So hearing in noise, or speech in noise discrimination, is the process of distinguishing speech from background noise, which allows for conversations in a noisy environment. Naturally, it declines after the age of 50, but then we see bigger declines in people with cognitive impairments. It can't, however, be measured by the pure tone audiogram, because there are people who do well on the pure tone audiogram, and yet they complain that they can't hear properly in real life. And now, a big part of speech in noise discrimination is the auditory working memory. This is the ability to remember sounds over a very short period of time. There is evidence to suggest that the hippocampus is involved in this task, and that the hippocampus is also an early target in dementia. Therefore, auditory working memory may be the link between hearing loss and dementia. So we hypothesized that hearing abilities correlates with speech in noise discrimination, which correlates with cognition. And a big part of that is the auditory working memory, which we believe may have something to do with dementia. So in our project, our research group has come up with our own new test of auditory working memory. So in terms of aims, firstly, we compared our new test to the conventional test of working memory. We then investigated the relationship between auditory working memory and central hearing, or speech in noise discrimination. Lastly, we explored potential predictors of auditory working memory. So over the summer, We recruited 36 participants in the early to midlife with normal hearing and no neurological problems. They were asked to do 4 tasks. Firstly, the pure tone audiogram, which tests their hearing in quiet. And secondly, hearing in noise. We also asked them to do the conventional test of working memory, and then our new test of auditory working memory. So this is just to show you what the speech in noise task is. Essentially, it simulates what real-world hearing in noise is like. So in this task, the participants were asked to put on the headphone, and they were played sentences over some background noise. These were sentences made up of 5 words, and on the computer screen, they were then asked to pick 1 word out from each of the 5 columns that corresponds to the sentences they could hear. So for example, it could be that Doris has 4 green flowers, or something like that. Now, in terms of cognitive tasks, we tested various aspects of their cognition. So for example, the phonological working memory, nonverbal reasoning, and so on. The digit span is a measure of phonological working memory. So in this task, I read some numbers out to the participants, and I asked them to repeat them in the forward and backward sequence. And despite the fact that these cognitive tasks have been used clinically for a very long time, and they are indeed currently the gold standard test in assessing someone's cognition, but yet there are limitations. So for example, there is a language component involved, and you may appreciate that not everyone's first language is English. These tests are also scored based on whether the participants can remember or hear certain words or sounds. So they are scored by yes, no, instead of using a continuous numerical spectrum. And these tests are also not easily repeatable, and they are user dependent. As a result of that, our research group has come up with our own new computerized test of auditory working memory. So in this task, basically I played them 2 sounds. I asked them to remember the first sound, and then when I played them the second sound, I asked them to match the pitch of the second sound to the first one. So this is just a schematic to illustrate this. At the bottom, I've got a little timeline, and here is just an indication of the pitch. Now here's the first sound. And then there will be a delay of a few seconds, followed by the second sound. So some of you may have noticed that there is a subtle difference in the pitch. Now at this point, using keyboard controls, the right arrow is to make the pitch higher, which will make it go this way, and the left arrow is to lower the pitch, making it go the other way. The idea of this task is to move the second sound, or the probe sound, as close to the first sound as possible. So it looks something like this. So at this point, once the participant is happy that they have made the right adjustments, they then press enter and move on to the rest of the test. And in total, there were 50 of these test sounds. Okay, so now coming on to results, we first looked at the relationship between our new test of auditory working memory with the conventional test of working memory, in which we found a statistically significant correlation. This suggests to us that our new test of auditory working memory correlates with the conventional gold standard tests that are being used clinically today. We then looked at the correlations of the old and the new test of working memory with speech in noise discrimination. We found that auditory working memory correlates significantly with speech in noise discrimination, whereas the conventional test of working memory did not show such a correlation. Now, this is in contrast to some larger studies that have indeed shown a correlation between the conventional test of working memory with speech in noise discrimination. The fact that in our study we couldn't elicit such a correlation is maybe because the conventional test of working memory is less sensitive of a test, therefore it requires a much bigger sample size to show a correlation. So in other words, we can say that perhaps our new test of auditory working memory has a higher power to it, and therefore it is more sensitive at predicting speech in noise discrimination. Lastly, we looked at the effect of playing musical instruments on auditory working memory, in which we found that the longer someone has been playing a musical instrument for, the better they performed on our auditory working memory task. And this is not just as simple as the fact that musicians are trained to remember sounds or pitches, because there are larger studies out there that have found that music training or music experience correlates with other forms of cognitive tasks that have nothing to do with music. So for example, the digispan, or remembering words or solving puzzles. So this suggests that playing musical instruments have a protective effect on more global cognitive abilities and not just auditory working memory alone. So just to summarize our findings, our new test of auditory working memory is more sensitive than the conventional test at predicting speech in noise discrimination. Therefore, our test may help predict the risk of future cognitive decline better. Also, playing musical instruments correlates significantly with auditory working memory. So music training may help reduce our risk of getting dementia in the future. Now, at this point, I would like to very briefly talk about my experience as a summer research student. In this project, I feel like I've gained a lot of new skills. So for example, data interpretation, data analysis, recruiting and conducting experiments on participants. And in preparation for today's presentation, I feel like I've honed my presentation skills. This project has also given me insight into what doing clinical research is like, and at the same time, it has given me a glimpse into the world of auditory neuroscience, which I find is way beyond me. So more importantly, I've made a lot of new friends, and I really enjoyed my time doing research. Sorry. Just one more slide to go. Last but not least, but very importantly, I would like to thank my supervisors, Dr. Meilin and Professor Tim Griffiths, my colleague Hannah, all the volunteers, Newcastle Biomedical Research Centre for funding this project, and the selection panel for allowing me today to present my work. And thank you all for listening.
Great. We've got time for a couple of questions if anyone has a question.
Can wearing a hearing aid decrease the risk of dementia?
Did everyone hear that?
So the question was, can wearing hearing aids decrease the risk of getting dementia? Am I right? Thanks for your question. So the honest answer is, I don't know. And first of all, because our project is based on the hypothesis that central hearing loss leads to dementia. And I know I didn't really have time to go into this in any detail just now, but apart from hearing in quiet and hearing in noise, there is peripheral hearing and central hearing. And in very layman terms, don't quote me on this, but peripheral hearing is how you transmit sounds from the outer world into your inner ears. And central hearing is how your brain then uses these signals or interprets these signals. And so from my study, I can only tell you that playing musical instruments may help with your central hearing, and therefore it may reduce your risk of getting dementia, but peripheral hearing aids concerns peripheral hearing. So I'm afraid from our study, I couldn't really answer your question.
Okay, one here, there's a mic just coming around.
Very interesting. What I was wondering about, when you present the noises to people, was there a difference between how different those initial noises were and the ability of the people to match them up? I was thinking, sort of listening to that, so but if you move the notes sort of closer, you hear what it wasn't. Does that replace your memory? The further apart they are.
I'll just go back to the slide.
Yeah.
Sorry. So I think it definitely confuses you. So the further apart the two pitches are, and when you're trying to adjust it, then you're like trying to hear a lot of sounds in the middle. And I think certainly the larger apart the two pitches, the more difficult it is.
Okay, and is there a correlation there sort of between your ability to remember and then the dementia risk?
And the honest answer is I don't know. Thanks for the question.
Okay, great, thank you. Thanks again. Okay, so next up this evening we have our Greenland expedition team who are going to be talking about investigating ice dynamics in West Greenland.
Hello everyone, my name's Anna and these are my teammates Isaac, Anya, Lucy and Hannah. We also have another teammate Katie who unfortunately couldn't be here tonight. We're all BSc Geography and Physical Geography students here at Newcastle University, and this summer we were funded to go on an expedition to West Greenland to study ice and glacial dynamics. So we went to a place called Kangerlussuaq, which is in West Greenland, and although being a town of only about 500 people in population, this is actually the international airport in Greenland, so it was much easier to get to than some other places in Greenland. It's also one of the most accessible areas of the Greenland ice sheet because there is a nearby road which goes to the ice sheet. This is actually one of the longest road systems in Greenland, and it's only about 60 kilometres long. You can see on the map in the middle here, these are our field sites marked in red on the ice sheet, and Kangerlussuaq is just a little bit off the map over here with the road heading up there. Other members of the university have stayed in Kangerlussuaq before, but they've actually camped nearer the ice sheet. However, because of climate change, there's been an increasing polar bear risk in this area, so we did not camp, and instead we stayed in the Kangerlussuaq International Science Support Site. This was very helpful in terms of giving us access to lab space while we were conducting our research, as well as hot showers, which were very nice after a long day in the field. It was also great to not get eaten by polar bears. We were really thankful for that. That picture on the right there is us getting our first sight of the ice sheet, which we were all extremely excited about. So we got to Kangerlussuaq by first flying to Copenhagen and then taking a connecting Air Greenland flight to Kangerlussuaq. Because these flights only leave once a day, we really didn't want to miss it, and therefore you can see us here asleep in Copenhagen Airport, which we spent about a total of 24 hours asleep there. Once we got to Kangerlussuaq, we needed to work out how to get from the town to the field site each day. Before we went, we were of the understanding that we would be able to get a lift. However, when we got there, we realised that this was not the case. And we actually considered both cycling and walking the 40 kilometres there and back each day, before realising that this was obviously not realistic, and instead hiring a car. There was a very steep learning curve with this vehicle. Here we are 30 minutes into our first field day, stuck in the sand on our way to the ice sheet. We look very happy in this photo because we didn't realise that we were going to be there for the next 2 hours trying to dig our car out of the sand, sort of Top Gear style, before someone finally came along the road and helped us dig ourselves out. But we did eventually get to the field site, and we did eventually get started on our research.
And so we chose to go to Greenland because to study the ice sheet because it plays important roles in both ocean and atmospheric systems worldwide. It's also currently undergoing rapid change as a result of the changing climate, which is shown by the left-hand image here with Greenland in the centre of the map, all highlighted in red, which shows that Greenland currently is undergoing very high temperatures compared to the average, with temperature anomalies of up to 5.5 degrees for the period of October to 2017, which is compared to the average from 1979 to 2015, so that's a very steep increase. It's important to understand the response of the ice sheet to these changes in temperature, particularly in terms of sea level rise, which, with current contributions of sea level rise from the Greenland ice sheet being shown in the right-hand image here. From this image, you can see the global reach of increased meltwater from Greenland having on sea levels globally, with the highest levels of sea level rise actually in the Southern Ocean, which is a result of complex gravitational effects from large-scale ice mass loss. While we were in Greenland, we also saw even greater elevated melt rates as a result of the European heat wave. So the melt extent from 2019 is shown here in the red line, which is greatly peaking above the average, particularly for the end of July and beginning of August here, with a massive spike up to 60% melt extent, which signifies the onset of the European heat wave.
So during our expedition, we spent 18 days in the field, and we all worked on slightly different projects. Me, Anya, and Hannah, we all worked on the actual ice itself, and we wanted to look at the key factors affecting the ice melt rate. So in order to do this, we installed 20 ablation stakes, which are the white plastic tubes you can see in the top photo. And to do this, we used a handheld ice drill. And the first time we actually tried to do this, we got the drill stuck in the ice for like half an hour. It was very stressful. But in the end, we managed to get all the stakes installed. And each day we would go around the stakes several times and measure how much the ice surface had melted against the ablation stake, and recorded this as the melt rate. We also used an anemometer, as you can see in this photo, to measure the air temperature and the wind speed in order to see if this impacted the melt at the different ablation states. We also took samples of the ice to determine if there was any biological activity in the ice surface. So Katie, our other team member who's not here today, she was studying glacial sediments, and she was working on the moraines, which are the buildup of sediment at the end of the ice. And she wanted to determine the rock morphology, so the shape of the rocks, and how this is influenced by the velocity and the speed of the ice. She also wanted to look at how the sediment travelled and whether weathering affected this.
So Anna's project was investigating the formation of debris bands within the ice. Rethwisch Glacier has very distinctive bands of ice, as you can see in this photo, and by investigating these we can see how they form and then from this we can see the dynamics of the Rethwisch Glacier. In reality this meant a lot of time hitting the ice with spades and ice axes, and even more time spent seeing the sediment. My project was looking at the carving mechanisms of the glacier, which is basically how much ice falls off. This is important because carving contributes greatly to sea level rise, so especially in the context of climate change, it's really important to study. I used a combination of time-lapse photography and data loggers measuring air temperature and water temperature. As you can see in this photo, I did a little remote control boat with a water temperature data logger attached to it. Our key findings were that carving, melt rate and lake temperature all greatly increased due to the 2019 melt season being so warm. You can see in this figure, this is all the carving events we saw during that time of field, which is much higher than normal. We found that a light surface debris cover also enhanced melt rate, whereas a great coverage can actually suppress it. The debris also contained biological material known as kryptonite, which can increase darkness and melting as well. The debris band we saw was caused by tectonic deformation within the ice. Away from our projects, we got the opportunity to help other researchers with their work. Here we are pictured with researchers from Loughborough University and the science team of Minnesota who took us out on these dinghies to study small lakes near the ice sheet, see how climate change is affecting them. And we also got the opportunity to help marine biologists from Nottingham University, took us on a boat trip 100 kilometres down the fjord to sample water, and we also helped them with their lab work. It's an amazing experience to see research firsthand, however it did seem like they just wanted to carry their heavy equipment and do the boring work.
We also saw a lot of wildlife while we were out there, as you can see here, a lot of reindeer who are not shy about coming close to you, a lot of muskox, which are the sort of hairy bison type things, a lot of Arctic hare, and thankfully no polar bears, although there was a polar bear skin on the wall of the local bar, which we did see. One of the biggest challenges with this trip was definitely the budget. Flights to Greenland are very expensive, as is accommodation and car hire when you get there. So our food budget suffered quite a lot. Food in Greenland is, as it turns out, also expensive, to the tune of about £3 for a loaf of bread. And when you're doing a lot of physical work, that really doesn't cut it. So we ended up eating a lot of rice, a lot of potatoes, and so many chickpeas that we actually bought out the town's supply of chickpeas before the next supply ship came in. And we found this out when another researcher tried to buy some off us. You can see a picture of me on the right there cutting a Mars bar into 4 pieces, because this was my only chocolate bar and I really wanted to stretch it out a bit longer. So when the researcher asked to buy our chickpeas, our price was, of course, in chocolate.
Whilst we were out in Greenland, we actually got interviewed by Channel 5 News. Because of such high melt rates in the 2019 summer season, because of the European heat wave, they came out and they interviewed us about climate change and what we think the global effects will be for places such as the UK, for example, an increase in the extreme weather events. We also took part in a charity 5K, as you can see in that photograph, in Kangerlussuaq, mainly because they said there was going to be a free barbecue at the end, which didn't end up happening, but we still really enjoyed it and it was very good to take part. And this photo was on our last day of fieldwork, where we got to explore the ice a bit and see some of the amazing landforms.
And so the key conclusions from our trip was that by analysing the data we collected in the field, we've all been able to answer our key research questions, which are informing our dissertation project. This fieldwork is also building on past student-led expeditions to Greenland, which took place in both 2008 and 2015. By carrying out this research, it's also given us amazing first-hand experience of conducting our own research in the field, which we've all really enjoyed, and these projects may also go on to inform future master's level research. Finally, we would like to say thank you to everyone who helped make this expedition possible, and in particular the funding bodies, the Newcastle University Expeditions Committee, the Royal Geographical Society, the Sheffield Church Burgesses Trust, and the Sheffield Grammar School Exhibition Foundation, without whom this exhibition simply wouldn't have been possible. Thank you very much for listening.
Okay, do we have any questions for our Greenland team?
How does the results that you've got in this expedition compare with results from previous years?
So we're still in the data analysis stage of most of our projects, but we are looking to compare them with the previous data, though we can expect that particularly the surface melt rates have increased as a result of the heat wave, though we are yet to finalise our results.
Another question? Yeah.
Thank you. It all looks very exciting. I'm a social scientist rather than a physical scientist, so my interest is, what is the barter rate for a bag of chickpeas in relation to chocolate? Fascinating opportunity to do a bit of social science.
We actually bargained pretty hard, and for 2 tins of chickpeas, we agreed on 4 chocolate bars. We received 8, but this might be because the researcher felt a bit guilty that we'd be carrying all his equipment. Chickpeas and hard work, basically.
Great. Thanks very much. Okay, so next up we have a research scholarship presentation. This will be given by Meilin Song, who's going to talk about learning from trees, and trees might be responsible for air quality.
Hi everyone, my name is Meilin Song. I study chemical engineering. My research is about learning from trees and how trees may be responsible for air quality. How do we measure the air quality? It depends on the number of particular matter. What's particular matter? It is a mixture of solid particles and liquid drops found in the air. It is also the most harmful form of the air pollution, including the viruses, bacteria, and metallic compounds. And particular matter has a wide range of sizes. Smaller sizes particles are more dangerous than the large-sized particles. Not only are small-sized particles presentation in the air for a long time, but they also interbody with our deep positions. I focus on 0.3 micrometers, also called PM 0.3. It would be like deposits in Olivera, causes respiratory diseases, reduce lungs function, and even lungs cancer. Why I'm interested in PM 0.3 rather than PM 0.1? Because the particle counter I use for my research projects, the smaller size can measure its PM 0.3, and we also can measure the air quality in this room. Just maybe waiting 25 seconds. So we can say there have two tubes, and this tube is the sensor for the temperature and for the humidity. And this tube, that's the air come in. Inside have a pond. This one is a camera, so we can take a picture or just make a video. So we also can say the smaller size particle always more than the large size of particles. That show me, okay, it's a PM 0.3 is less than 6,000, so it's a good air quality because they're still in the right green area. If the air quality is poor, this counter will go away. And my aim was to find the relationship between photosynthesis reaction and the number of particles. What did I do in Newcastle? I chose different locations between Central Garden and the old libraries with a different plant. So I measured the number of particles with 2-hour intervals. So the trends show me very similar with the different locations. So that show me between around the 11 a.m. and the 3 p.m. is a large number of particles of PM 0.3. So this means it's a good air quality rather than in the morning or the evening, with less human activities. So, because I want to know the photosynthesis with the number of particles, because we know for the photosynthesis reaction happen must have the sunlight. So I do another part for the nighttime. I'm interested in the air quality change during the nighttime. And also the figure 1 show me what different locations give very similar trends. So I measured the figure 2 in my room by opening the window to simulate the environment in my room as the same as the outside. So that shows me the same things as the figure 1. So between the 11 a.m. and 3 p.m. is the large number of particles, and during the nighttime there have the more particles in the poor air quality. So can we say plant or photosynthesis reaction will improve air quality? No, because there have so many uncontrolled factors. For example, the speed of wind or temperature in Washington. Before I do my second part of reaction, I go back to my hometown, Tianjin, which is close to the capital of China, Beijing. So you can say that's the Pengbi is Beijing, Pengsi is Tianjin. That's like Darlington and Newcastle. So what I did in my hometown, the control experiments was set up. 4 same-size boxes was used, and 3 different plants in each box and 1 empty box as a control group. And I put a light incense stick into each box for around 6 seconds to show me the number of PM0.3 produced in each box are the same, it's 10 power to 6. And I'm waiting around maybe 1 hour to wait the particle in the box to complete distributions. And also I measured the environmental air quality in my hometown, because we always think about in Tianjin or in Beijing is a high air pollution. So this one is for 3 days. So we can say the first days show me the same trend as the Newcastle around noon, so without good air quality. And during the night, the air quality become too poor. So it's really similar with Newcastle. And also, we can say for the normal day, maybe the first day and second day, that show me maybe the lowest pound is 25,000. And if we remember the figure 1 in Newcastle, that also almost around 25,000 of the PM0.3. So I can say the air quality in Tianjin is similar in Newcastle, and Tianjin not is a high air pollution city anymore. Maybe before 10 years it's true. So this one is for my control experiment. So we can say for the empty box there is nothing. So I interest the time take from the number of PM0.3 decreases from 10 power to 6 to 25,000. So this blue line show me 25,000 at here. So almost that's maybe 20 or 21 hours like this. But without plant, we can say 25,000 at here, almost just 12 hours. Almost just 12 hours. And if the box without plant, the particles decreases quickly than empty box. Whatever which plant, because they show me same, okay, almost 12 hour, almost 12 hour. So I can say the plant will improve air quality. But you also found me why my figure that have some peak during at this area have peak, or this one because my box not 100% sealed. So I also to compare which plant is with high efficiency. So I just do a simple calculation. I calculate the total surface area, and I calculate the time the PM0.3 to decreases. Then I just divide by. So I found the monastery has the highest efficiency as per hour per centimeter squares. So for my future work, the box should be improved by 100, so complete sealed box, and a fan should be 8 in each box to simulate the real environment, because I have some wind, but in my box there are no wind, so my particles decrease so quickly. And also, I hope the electron microscope should be used to see the structure change during the sunlight and without sunlight. Try to find the fundamental mechanism about the plant or the leaves absorb the particles. That's all. Thank you for your listen. Do you have any question?
Should we be planting evergreens rather than deciduous trees? Because generally trees evict leaves, don't they? And
Yeah, in the
become
winter.
less tree-like.
In the winter, I mean, but I think maybe that's during the winter, depends on the city actually. In Newcastle, the trees still have the leaf dry, but in my hometown, there are no leaves anymore in winter, the snow.
And just a question here.
Do you know what the composition of those particles are? Particularly, sort of, does it change during that sort of daily cycle? You've got sort of those big peaks around about sort of 6 o'clock in the morning to 9 o'clock. Is that sort of all coming from diesel?
I'm not really sure. I guess some reason because the plant and because during the morning the sunlight is not really strong, and also we have the temperature in ocean. So this one is if the temperature in the air is higher than the temperature on the ground, that will become a thermal layer. So the particle cannot to run away. So that's like something to cover. So just keep in the same area almost. But I'm not really sure because I didn't do this one. Sorry.
Okay, thank you very much. Okay, so next we're going to be heading to Peru for our talk by our expedition team who are investigating the flora and fauna of Manu National Park.
Hello everyone, good evening. My name is Lydia. I'm a third-year ecology and wildlife conservation student.
Good evening, my name is Alex Lowe. I'm a third-year BSc Applied Plant Science student.
Hi, I'm Jennifer. I'm a third-year ecology and conservation biology student. And we also went with Lydia, who can't be here tonight. But I led the Peru team investigating the flora and fauna of Manu National Park. So we went to Cocha Cashu Biological Station, which is in the southeast of Peru, and it's situated in the heart of Manu National Park, so it's well past the tourist zone. And this place is really special because it hasn't been influenced by man, so there's been no hunting, there's been no deforestation, and there's been no fragmentation. So this is really important because we were primarily interested in the key ecological processes that occur there, and the human influence hasn't impacted any of these. So to answer our aim, we split the aim up into 4 projects, which we're going to talk about today.
So I'm going to talk a little bit about the journey to Cocha Cashu, which you probably guessed took quite a while, 5 days in total. So first we had to get to Peru itself, so we flew from Manchester Airport to Amsterdam and then to the capital of Peru, Lima. We then ran through the airport itself to catch another flight to an Inca city called Cusco, which is situated 11,000 feet in the Andes, where we spent a hasty one night, caught up on some sleep. Then got a van over the Andes itself, up to 14,000 feet, and then down into the Amazon basin, where we stayed in a lovely little boat town called Atalaya for the evening. The next morning when we woke up, we were completely immersed in the rainforest for the first time, which you probably guessed is quite an incredible experience. We boarded a boat with some other researchers who were heading to the station, went down the Rio Manu, went to a little village called Boku Manu, and then spent the night in a little research station called Limonel, where we had to sign into the national park as no tourists are allowed to go past that point. Spent the night then, and then we took another boat the next day up a continuation along the river to Cocha Cashu station itself, which is quite nice to have a shower for the first time in 4 days.
So we lived at the station for about 6 weeks, and we lived on platforms like this. This is me and Laura's lovely platform, which is slightly raised from the ground, and we had a tarpaulin, but other than that we were pretty much in the forest, a little bit away from the camp. The camp was in a cleared area. There were 3 main buildings: the changing room, the dining room where we had like all of our food and there were kitchens there, and the of this area where they had a library, laboratories, and we all had these adorable little cubby rooms where we had a lot of team meetings and chatted in the evening. So we had to do our own laundry, which became quite a social thing, but sometimes it was annoying that it would take like an hour to scrub all of your clothes after you've been doing field work. We'd get buckets from the Cocha, avoiding the giant otters and the caiman, bring them back and scrub our clothes, try and get it clean, but they would then be clean. And we would also eat with all of the other researchers, which was really lovely. When food was ready, they would call out, "Rancho listo," and we would literally run there. And it soon became our favourite phrase.
So there's a lot of dangers associated with living in the rainforest, some of which you can probably think of yourself in regards to the big cats, the puma and the jaguar that occupy the area, the venomous snakes, some of the vipers, little ants that if you get bitten by them it feels like you've been shot by a bullet and it lasts for 24 hours. But one of the things we didn't really expect to be too dangerous during our time there was actually the danger of falling trees, which occurs quite frequently in the rainforest. During our time there, a huge tree fell on a fairly gusty day, no windier than you would get here in Newcastle, which actually crushed one of the platforms that our research, some of the other researchers were staying at. Fortunately, they weren't occupying the tents at the time, as you can imagine it would be a bit of a disaster if they were. One of the other things we weren't too concerned about when we got there, but it did, I don't know, become something we were quite aware of, was the fact that there is a lot of uncontacted people which live within Manu National Park. So they're indigenous people which have no contact at all with the outside world. There was this reassuring translation sign which was situated in the dining room of just some phrases that you should memorise in case you come across some uncontacted people when you're in the forest, such as brother, which is Namole, and please don't kill me, which is you are comfortable. Fortunately, nothing of the such happened whilst we were there, but it was something that you did have on your mind when you were wandering through the forest.
So our first project, which I led, was factors affecting the habitat use of jaguars and their prey. So I really like big cats, so that's why I picked this topic. But other than that, jaguars are keystone species, which means they regulate the population base, which then in turn affects how much vegetation is eaten by the prey. So this keeps the ecosystem together. And also I was in talks with people from San Diego Zoo, Peru, who own the station, and they were really interested in my project, and we decided to do a collaboration. So they set up some paired traps and I set up my own, and we wanted to set up a more long-term monitoring program of the mammals there. And also no one knew how many individual jaguars were present at the station, or no one had attempted to identify any individuals. So I set up 20 camera traps across the site in a grid, and one of the factors that could be measured that could affect the habitat use was canopy density, but I'll talk about that later because it was involved in another project. So we actually got quite a few images of jaguars from the camera traps. This is just 3 examples. So to identify individual jaguars, you compare the unique spot patterns on their coat. So this is the female that was present at the station, and you can see that she has this smiley face on her leg, which you can compare it to in these other images. And she also has this curly C shape on her forehead as well. And we also got some videos. So this is just 500 meters away from camp. Yeah, right at the port. And we also discovered that she has 2 cubs. They were also caught on subsequent camera traps which were further down this trail.
Okay, so my project was studying insect delivery. It may not be exciting as looking at jaguars, but it's a really important process. Delivery, for those of you who aren't aware, is the process in which insect herbivores consume folio leaf material, and is very important in the rainforest, as you can imagine, because there are millions of insect species themselves. It can be a process which can regulate plant growth as well. So it was what I wanted to study while I was there. So to do this, I collected 6 leaves from 6 plots which I had placed in the forest, I would then measure the percentage of herbivory damage using a handy little BioLeaf app, which would give me herbivory damage percentage, seeing the areas which are indicated in red on the right, and the leaf area index. I would also categorise the leaves via what type of herbivory was present there, such as leaf miners or hole feeding. And this is really important because it can give me a gauge of what sort of insect communities are present in those habitats, which can then lead on to further research of how predators are distributed within the rainforest, and also can be influenced by changes in climate change and how that will affect predator and prey relationships.
So our third project was led by Lydia, and we decided to focus on strangler fig tree distribution. So if you're not sure what a strangler fig tree is, there's an example on the board, but essentially it's like a parasite tree that grows on top of another host tree, and it grows stronger roots around this tree and eventually strangles the tree to death, which hence the name strangler fig tree. And in this example, the host tree has already died, and there's just the roots of the strangler fig tree left. So you could actually walk through this tree, which is pretty cool. And fig trees are also considered a keystone species because they provide a huge food resource for many of the mammals during the dry season when other food items are unavailable. So to measure the distribution of the fig trees, we walked the trails and logged their location with a GPS. And then we also measured the diameter and height of the host trees and the strength of the strangler fig. And we also measured the canopy density, which is how dense the vegetation is. So to do this, we used a compass and measured a 20 by 20 meter plot around each tree. And then we had a special camera, which was fitted with a circular lens. And then you go to each point in the plot and then hold the camera on top of your head and point it to the sky and take pictures. So you end up walking around the forest with a camera on top of your head, which looks a bit strange, but you get images like this one on the bottom right. And then you analyze these pictures to get a measure of canopy density.
My project was on palm trees. Palm trees are a major component of neotropical forest. At Cocha Cashu, palm trees make up 3 of the 5 main tree species that make up the forest, which means that they have like a great effect on how the forest is. I wanted to look at whether the distribution of palm trees and the species composition of palm trees was different in different areas of the forest. At Cocha Cashu, because it's near a river, which is actually bordering this whole map that you can see here, the forest has different stages of growth. These can also be called successional stages. So when the river was previously covering this area of forest, it flooded it, and so when the river goes back to being where it is now, the forest reclaims it, but it's not necessarily the same type of forest as mature forest. So my 3 plots are on the board, the little red dots. They were randomly generated points in different habitats, and at each of these 3 dots I had a 10 by 100 meter quadrat, and in the quadrat I measured the positioning of every palm tree and every forest tree above 10 centimeters DBH. I also measured the height of all the trees, and I measured whether the palm trees were mature or not. With palm trees, it's actually quite easy to tell whether they're mature or not because the flowering structures often stay around on the ground, or you can find seeds or something. Something that's really fun is that if you're looking at not a canopy palm tree but an understory palm tree, you can actually feel a scar from where it had once flowered if you go along the stem. Yeah.
So moving forwards, so we did a collaboration with San Diego Zoo with our camera traps, and we exchanged data, and we're going to use that data to apply for a grant to buy some permanent camera traps to set up at the station to continue the mammal monitoring program. And also there was a visiting school group from one of the local communities, which I did a presentation for, and they were telling me about the mammals at their communities, and they were also telling me about the human-wildlife conflict that happens there, so it got me thinking, and I'm developing another question. So hopefully next summer I'm going to go back to Manu and compare the mammal communities within the other villages within Manu.
For
apart from working, we had so much fun. I mean, we were in the jungle. We went out on the Cocha in canoes that were built by the local Machigenga people, and we explored the beaches looking for paw prints. And we also went exploring through the jungle. This was one of our favourite days, I would say, where we went to this amazing place which we found Playa Bonita, which is kind of in the name, and was also the picture on the first slide. And so we had a great time. We had chances to do a lot of fun things too.
We also had the opportunity to help out with other researchers who were present at the station. So this is just one of the examples. But I went on a night walk with one of the researchers, and I collected these giant grasshoppers and cockroaches and had to hold every single one of them and put them into individual tubes, whilst avoiding all the poisonous spiders and tarantulas that were hiding under the leaf litter. But it was really fun. And, and we also asked the chef if we could bake a cake for dessert one night, and everyone really enjoyed it, so it was good.
And we'd also just like to thank everyone who helped with the expedition, especially the expeditions committee at Newcastle University, and a special thanks to Dr. Marvin and Philipp Rethwisch from the amazing audience, and Dr. Gordon Thorpe as well for aiding us, and without whom I don't think the expedition could have gone ahead.
Thank you.
Great. Do we have any questions for our Peru team?
Could you live stream your cameras could you live stream your Jaguar cameras and see, sleep in your tent and see whether the Jaguar is 5 meters away or not?
Unfortunately not, no. I had to check the cameras every 2 weeks on a rota just to check the SD cards and swap them. And then I took the SD card back to the office and then downloaded the images from there. And then I was just pretty much sat in the dark going through thousands of images, yeah.
Would you want to know how close they go?
Oh, yeah, we had a competition of who got within the closest time of being present where the Jaguar was as well. So I had a little leader table. I think the closest someone got was 40 minutes between the Jaguar, yeah.
Okay, great. Thanks very much. Oh, one quick question.
I'll just check there. How do you count all the trees in the 10 by 100 meter corner?
A lot of time. A lot of time, and a lot of help from everyone else. I mean, it was a different. We helped with each other's projects, and yes, it was a lot of time. The other thing is that I discounted the saplings below 30 centimeters. I didn't measure because otherwise it would have been impossible. And the forest trees in different areas of the forest, I would say in the mature forest, you'd get massive ones. The diameter would be crazy, but then you would either have massive ones or very few ones that were bigger than 10 centimeters DBH in the late stage successional forest. And in the primary forest, I think I found 2. So.
Lots of dedication. Great, thanks very much. Okay, so our final presentation this evening is going to be given by our Russian expedition team, who are going to be telling us about their Sion Mountain expedition.
Hello, I'm David, this is James, this is Meg, and we went to way out in the Far East in Siberia to the Sine Mountains. We also had another group member, Harry, who isn't with us tonight. So we went out there to reconstruct past environmental conditions. So reconstructing past environmental conditions is really important because knowing what temperatures were like in the past, for example, helps us make predictions of what's going to happen with climate change in the future. And in Russia in general, there's very few studies into climate in the past, and where we went to Sine Mountains, there's even fewer. So our work here was really important. So what we did was we cored a series of lakes to get sediment cores from them, and we looked at the microfossils within these cores to be able to work out what conditions were like in the past. So this trip wouldn't have been possible without our Russian counterparts, which we've got in the far left picture. We've got Sergei, our driver, on the left-hand side, Elena and Misha in the middle, who conducted their own research but helped with all the logistics of the trip. And then in the centre, Ivan and Sasha, who are massive experts in the reconstructing past environments field and joined us on our expedition, conducted their own work but also helped with ours, helped set up the wild camp and things. And then on the right-hand side, we've got our hitchhiker, Alexander, who joined us on our journey there and then came along the whole trip, wild camp with us, came to all of our field days, didn't speak a word of English, but he picked wild rhubarb and stuff for us to try, so he's just a nice guy.
Okay, so our journey started from Heathrow and we flew to Irkutsk with a 14-hour layover in Moscow. So we got to go into the city and look at the Red Square and the Kremlin. So we arrived in Irkutsk and had a few days to acclimatise to the time difference. So our Russian counterpart showed us around the institute, showed us where they keep their lake cores, which is in an old nuclear bomb shelter. And then we had an explore around the city and got to go to Lake Baikal, which is the largest freshwater lake in the world, holds about 20% of the world's freshwater. So after a few days of chilling out a bit, we travelled to the field site. So we travelled on the first day by minibus, which took about 8 hours. Ended up in Orlik, which was a small satellite mountain town, which we got there and realised that we were going to have Wi-Fi for the last night, and then everything just cut off. We didn't even have lights anymore. So it was our last night in a bed, but wasn't as great as what it could have been. So then the next day we got in a, you can see in the middle photo there, it was an old army off-road truck, which was a lot bumpier than what I think we were expecting. We got quite a lot of airtime. So it took about 10 it took about 10 hours to get there, but then by the time we did get there, it was getting dark. We got to go in the hot springs and then slept on a wooden plank in our sleeping bags.
So the last bit of our trek to get to the field site, we had to do a 17-kilometre walk with a 900-metre climb to get from Key to Goal to the campsite, so that's along the red dotted line you can see in the map. With us on our trip, we had some guides from Key to Goal and their horses who helped carry all of our equipment. The weather conditions were pretty terrible. It rained the whole time, the rivers were really high. We got Harry battling his way through a river on the right-hand side. And the weather also led to our plans for camping to change. We were going to move the campsite each day from lake to lake, but we ended up having a fixed campsite, so we didn't want to move it around and get it soaked. And also, sadly, Meg got ill, so we wanted to keep her in one place. So here's some shots of the campsite. Me and James looking miserable because we're wet in the bottom left. All the expeditions team at the top picture. And then on the right-hand side, we've got our little bit of tarpaulin that we always hid under from the rain, and our three Russian counterparts having a go at an English crossword. Not doing very well. So the first lake we went to was Lake Kaskadno 1. And for each lake we took both a short and a long core. So the process for taking a lake core is we'd inflate these boats that you can see in the centre, roll out into the middle of the lake to the deepest point, so in this case the lake was 6 metres deep. And then you take the core, which is the top left-hand photo, and attach a long piece of tube to it, and then lower it off the side of the boat with rope, and then use the weight on the end of the core basically to shove the piece of tube into the sediment at the bottom of the lake, and then pull that up, and you've got your tube of sediment that you take out and to analyse later.
So we also went to a second lake, which was called Lake Cocha, and this lake was significantly deeper than the first lake we went to. So Lake Escambay 1 was about 6 metres deep, and this lake was about 40 metres deep. And it took us about 2 hours to walk there, because as you can see in the middle right-hand photo, the whole of the valley floor was covered in a large lava flow from the nearby volcano, which was thankfully gone whilst we were there. But this didn't mean that because it was still raining, it was very slippery on the part of the eagles. So these are some pictures from our journey back. It's worth saying that whilst we were at the field site, it rained the whole time, but when we left, it suddenly became nice and sunny, which is typical. But because of all the rain, I remember the journey back was quite a lot slower because the ground became waterlogged, so we had to actually get out of the Euro car and make our way on foot for a little bit whilst the car navigated around. We did also have the chance to pick up a school trip, which had been going around the local shrines and picking up all of the vodka bottles which were deposited. Because when you visit shrines, you're supposed to make an offering, usually a full vodka, which involves you pouring some vodka into a cup, putting some on the ground as a sign of goodwill, then flicking some over your shoulder, and then down the rest. And so we also picked up a nice dog who belonged to the driver of our Euro car. However, although the final picture in the bottom right looks lovely, that's actually where we stopped because the dog was sick. All the bumpy journey. And so when we got back to the Institute of Geochemistry, we started our lab work, and this involved cutting the core that we'd taken in half, and the Russians kept one half of the core, and we took the other half for our analysis, and we then cleaned that and sliced it up 1 centimetre intervals, and put that in Ziploc bags to bring back to the UK.
So my research is on chironomids, which are non-biting midges. So in the far right hand side, you can see a chironomid. I look at not when it's in fly form, but when it's a larvae. And its head capsule that it's circled is made chitin, so it gets preserved as a fossil, so I can use this. So my study is reconstructing Holocene temperatures using these chironomids, the Holocene being the amount of time since the last ice age, which is about 12,000 years. And as I've said before, this is really important because knowing what temperature was like in the past helps us predict future climate change. And basically how this process works is we use this core that we took from Lake Escambay 1, and the deeper you look down the core, the older that is in time because sediment at the bottom of the lake builds up over time. And then chironomids are really environmentally sensitive, so certain species only live at very specific temperatures. So at a certain depth, I'll identify as many chironomids as possible, and then based on what I've found, I can interpret what temperature is likely to be at that certain depth and therefore that certain point in time, and I can create a graph showing how temperatures change through time in the past.
So my research is also on chironomids, like David's, but I'm looking at a much shorter time scale. So we often think of climate change as happening over a long period of time, but in actual fact it fluctuates quite rapidly. So in the last 1,500 years, approximately, there's been 2 key periods which I'm focusing on for my research, which are the medieval warming period. So there's evidence that Vikings inhabited Greenland and farmed there. And then the Little Ice Age, which there's evidence of people ice skating on the Thames for. So I'm looking at how Russia has responded to these changes as well, if they actually happened at all. I also didn't, I didn't leave the tent for a week either, so I thought I'd put some photos on here of what my experience was like, as I didn't actually get to visit the field site. So I got absolutely bitten to death, and then whenever it did stop raining,
we had to get our mozzie nets out because they just absolutely appeared from nowhere. And then I spent quite a lot of the week drying people's clothes in my tent whilst they were on the field work. And then on the last day, as Jim said, the sun came out and something landed on me that wasn't a mosquito, so I took a photo of it.
So my research is into diatoms, which are single-celled algae, and I'm looking at the way that lakes or our lake in Russia responded to ecological forcing. So there have been widespread changes in Northern Hemisphere lakes, particularly in Arctic and alpine regions across North America and Europe. But because of the lack of studies in the South Mountains and Russia in general, we don't actually know how Russian lakes have responded. So there have been 2 studies using diatoms in the South Mountains, and one has followed the global trend and one doesn't. So my study is aiming to see whether mine does follow it or doesn't. So Harry's research is using spheroidal carbonaceous particles, which are just soot from industrial pollution, essentially. And he'll be comparing 2 lakes, one from close to Irkutsk, which is where we were staying when we arrived, and what the core from Lake Escapa 1. And he'll be able to tell from this, because the particles are only produced from human pollution, he'll be able to tell when human pollution started, and also examine to what extent the Cascade 1 area can be considered pristine. And finally, we did see a bear whilst we were there, but we didn't realise it was there until one of the guides fired a warning shot. Then we all turned around and saw a bear running back up the valley. So unfortunately, we were never in any real danger, but hopefully you can see it. It was quite far away, unfortunately.
So to conclude, all of our research projects take loads of microscope work. So we haven't actually finished all the data analysis yet, so we have very few scientific conclusions. But we've already got a lot out of it. We successfully collected our cores for our dissertations. Because we were with our Russian counterparts, we felt we were able to really immerse ourselves in really interesting Russian culture. And it was a pretty nice place to go. And for future work, as I've mentioned before, there's a massive gap in knowledge in Russia and in the Sire Mountains for past temperature and environment reconstructions. So there's loads of other lakes in that area that could be looked into the same thing, and there's lots of other types of fossils aside from chironomids and diatoms and SCPs that could be used for similar types of research.
And finally, we'd just like to say a big thank you to all of our funders and the people who've given us amazing advice. So Dr. Mike van Heidenbroek was absolutely invaluable, and again, all our Russian counterparts as well.
Great, thank you very much. Any quick questions? Yeah.
Oh, brilliant, thanks. Thanks for that. I was thinking, sort of, did you acclimatise yourself by going to Kielder for the midges? That's not my question. My question is, the sedimentation in these lakes is going to be different. Yes, it's not going to be constant. So how do you know what the time is when you're looking at your timelines?
Yeah, so I forgot to mention that. We've sent off samples to get radiocarbon dated that we'll get back in the new year, so we haven't got those back yet, but they'll tell us, well, not the exact dates, but with an error margin, what dates each depth relates to.
I just had a question. I wondered how we got on with the Russian crossword.
They didn't have a Russian crossword. They had to go at the English. They didn't have one for us to have a go at. But we did learn some Russian words, but they're basically all to do with food or cutlery, because whenever we had meal times, like, they couldn't speak English too well, and we knew no Russian. So we sort of just point at a fork and they'd say what that was, or potato, which is kartoshka, and stuff like that. So yeah, got niche Russian language knowledge.
Great, thanks very much.
Okay.
Okay, so that brings us to the end of our presentations for this evening. You've obviously seen what a diverse range of topics our students have covered. These are just a few examples of what's been going on over the summer. Also, for most of them, this is the first time they've presented, so I think you'll agree with me they did an amazing job. I'd like to thank them again very much for their presentation.